Good afternoon, everyone. Thank you for joining us virtually for today's seminar. I have the great pleasure of introducing our speaker today, Dr. Michelle Wille. Dr. Wille works on scales crossing research in ecology and evolution using field techniques, lab, and computational work to study the dynamics of avian influenza. Dr. Wille earned her bachelor's in science with honors in biology, followed by a master's in biology from Memorial University in Newfoundland, Canada. She then completed her PhD in the Center for Ecology and Evolution in Microbial Model Systems at Linnaeus University in Sweden, followed by a postdoc at Zoonosis Science Center at Uppsala University in Sweden, the WHO Collaborating Center for Reference and Research on Influenza at the Peter Doherty Institute for Infection and Immunity in Australia, and the Sydney Institute for Infectious Diseases at the University of Sydney. She is now a senior research fellow at the Center for Pathogen Genomics, Department of Microbiology and Immunology, Peter Doherty Institute of Infection and Immunity in Melbourne, studying ecology and evolution of avian influenza A in wild birds and revealing the ecological factors driving the avian virome diversity and composition. We eagerly anticipate her presentation today and the valuable insights she will share. So without further ado, please please join me in welcoming our speaker, Dr. Michelle Wille.
Thanks for that introduction. I feel like you made me sound like I go everywhere, but I go everywhere, so that's quite fun. So I feel like you stole my thunder a little bit because I definitely got asked to introduce myself before I started, and that's what I was going to say. So I'm going to talk to you about avian influenza a little bit across scales. And as Steph said, I have had an interesting career across kind of diverse places, thinking about lots of different things. And so I won't spend a lot of time on this, but just thought to share this. So I'm Canadian, but living in Australia at the moment, and have spent most of my time working on avian diseases across various scales and sometimes visiting the same topic across multiple scales or sort of through different lenses. So when I was living in Newfoundland, which is the eastern part of Canada, it's quite a really interesting place and really rich in seabirds. And so I really thought about questions like avian influenza in seabirds along these sort of continental margins and how that system is working. I will just highlight that my honors I actually worked on a bacterial disease outbreak caused by a bacteria called Pasteurella maltosida. So I do have the capacity to work on bacteria, as many of my colleagues often forget, though it's been a while. One of the other really fun things about the work I do is I get to do field work in lots of really cool places. So I just put a little map there with some of the fun places I got to visit and work at across some of my positions. So yeah, that was Canada. Then I moved to Europe, worked in Sweden for a while, and really started thinking about avian influenza in great depth. So within one population of birds, how are these viruses maintained over kind of decadal scales? And ducks are really nice creatures to work with. I grew a great affinity for ducks. So my first postdoc, I worked more with experimental infections, which was really interesting, and a deviation from natural systems. But I decided that I much preferred to work on natural systems. And then I moved to Australia and did a chunk of work on avian influenza, but actually also did quite a lot of work on virome scale research, so trying to understand everything that's going on in birds. And I think a lot of the things that I've done over the course of my career has been woven into this. Of course, not everything, but many things. And so what I've done is I've tried to take all the avian influenza work that I do and just really weave it through the different lens of different scales. I really like this framework and I got a bit overexcited, so I think I crammed more into this presentation than I normally would. So hopefully it's interesting and not too long. So I really wanted to start at the scale of multi-host one virus systems and looking at the global scale, okay? So before we do that, I just want to briefly introduce avian influenza and what it is for those who are perhaps not familiar. So avian influenza is caused by a virus, influenza A virus, and this virus is very, very diverse. So on the outside of this virus are sort of two lollipop proteins called the HA and the NA. So from COVID, we all learned about spike proteins. In influenza, essentially, the spike is broken into two. And so we need two proteins to do the job that just spike does. And then these are highly diverse, okay? So within the HA, there are 16 different types that are found in birds. So if you take these and you try to align them, they'll only have like 60% similarity. So far more diverse than COVID at all, far more diverse than variants. Like, we're talking on quite a different level. So 16 HAs, 9 NAs, and we categorize these in kind of combinations. So if you've been reading the news, what's in dairy cows at the moment is H5N1. And essentially, in wild birds, almost every combination has been found. The other way we categorize these viruses are through pathotypes. So we have low pathogenic or low pathogenicity and high pathogenic or high pathogenicity avian influenza. The vast majority of avian influenza out there are low pathogenicity. Okay, so almost all those HA-NA subtypes are low path. They're very common in wild birds. They cause no clinical disease in wild birds. They're likely co-evolved with wild birds over this sort of scale of bird evolution. This is in contrast to highly pathogenic avian influenza. So essentially, subtypes H5 and H7 would enter poultry production systems. And within these poultry production systems, they have genetic changes that occur in the HA as they replicate and move through these big barns. We see the insertion of some amino acids, and then these viruses become highly pathogenic, okay? They have very high virulence. They cause really substantial outbreaks in poultry, significant morbidity and mortality. And these days, they're actually also associated with significant morbidity and mortality in wild birds. And I'm going to be talking both about low path and high path in this presentation. But at the global scale, I thought I'd talk about high path because that is actually the topic of today. So in 1996, a highly pathogenic avian influenza virus emerged in China. It's called the Goose Guangdong lineage of the virus. And unlike all the other iterations of high path that have emerged, this one was never stamped out. It was never controlled. And it has essentially become endemic in birds. So sort of between 1996 and probably 2014, this virus was largely contained to Africa, to Asia and some parts of Africa. We had a bit of a step change in epidemiology in 2014, where we started seeing outbreaks occurring in Europe. So this virus emerged in wild birds. And then really the wheel fell off the cart in 2020 when we had another sort of evolutionary change and this virus spread around the world. And so I just want to show you sort of the stark contrast. So this is notification data from April 2020 to September 2021. So each dot is just a notification. It could be one bird. It could be a million birds in a poultry house. And we've just divided the colors here by poultry and wild birds. So this had been the situation for some years. And then this happened, okay? So the virus spread over the Atlantic to North America, subsequently also over the Pacific, causing mass mortalities, significant outbreaks in Canada, the US, and Mexico. Approximately 1 year after entering North America, the virus entered South America. There were at least 3 introduction events. The virus traveled the 6,000-kilometer spine of South America in about 6 months. It has been causing massive, massive levels of mortality. More than 600,000 wild birds have died in South America. We've also seen more than 40,000 pinnipeds have died from this in South America. Approximately 1 year after entering South America, the virus entered the Antarctic region. We've had outbreaks in South Georgia Island, as well as the Falklands, and the first detections occurred on the Antarctic Peninsula somewhere around February or March. So Oceania, where I am, is the only part of the planet where this virus is absent. So kind of going back to what happened that caused this massive explosion, there were a few things, but for about the 6 years previous to this change, we saw mostly this H5N8 subtype combination and the emergence of this terrible lineage name. We love to do this with influenza. We give them these awful names. And we had this thing called 2.3.4.4, which had subdivided into lots of sublineages, sort of A through H. And in different parts of the world, sort of these different lineages were circulating. But there was a really strong selection event that happened here in kind of what we your summer 2021, and the strong emergence of lineage 2344B, and that's what we're seeing everywhere in the world now, and H5 returning to its old foe, old friend, N1. Okay, and so this sort of combination has now outcompeted everything. It's outcompeted everything that was in Africa. It's outcompeted everything that was in Europe. And it's progressively competing out what has been circulating in Asia for decades. So big genetic change, causing really substantial changes. This has also changed the kinds of birds that it's affecting. So really prior to this, on the left-hand part, we saw that this virus was mostly affecting waterfowl, in a few reports in the literature of big outbreaks and things like teals. But and affecting some raptors there in orange, because obviously raptors are eating the dead teals. And there used to be a really strong seasonal trend where there was not a lot of activity in the summertime. But since that change, it's really emerged into this green color here, which are colony breeding seabird species. And this has really had a huge and detrimental impact on seabirds with population and species level effects. So, for example, 16% of all the sandwich terns in Europe died within a 2 or 3 month period. 40% of all the Peruvian pelicans in Peru similarly died within just a few months. So huge, huge, huge impacts. So sort of based on the chart I showed you before, it's not sort of unexpected that we're seeing a really big increase in the avian diversity that are being affected. So that reddish colored line matches with the axis on the left, and that is sort of the number, just the sort of species diversity plotted. And then what we've done on the axis on the right is showing you that in bars. And so, for example, in light green, there are the Schradriforms, and the size of the bar is the number of different Schradriform species that are affected. So you can really see that everything has increased since 2020, and we're just seeing a much, much bigger diversity of birds. And this data is until 2023. It has only increased since. And of course, as I said before, it has resulted in some really substantial mass mortality events. So in a paper that came out last year, we really looked at some of them to look at this sort of species level effect. And so at Bass Rock in the UK, which has this incredible gannet colony, there's been a 71% reduction in gannets that have been found there. And, 20,000 South American sea lions have died in Peru and Chile. The numbers just are really very staggering here. This is a huge conservation issue for birds, but also for mammals. So, this is called avian influenza because it primarily affects birds. It primarily binds to these avian type receptors, but we're seeing lots and lots of cases of mammals. Everywhere mammals are being tested, they're turning up positive, which means that on this map where there are no mammals, it doesn't mean there's no mammalian infections. They're just not being tested. In general, we're seeing cases in predatory and scavenging mammals. So if you imagine all those dead birds on the landscape, it is an all-day buffet, and foxes and all kinds of things are eating these dead birds, and they themselves are becoming infected through this route. The exception, of course, being these massive pinniped outbreaks, where there is strong speculation of mammal-to-mammal transmission, because essentially these outbreaks have kind of traveled and moved. And so in South America, we started seeing sea lions in Peru and then Chile, and then it kind of continued south through Chile and back north up through Argentina, and it's continued into Brazil. So there's clear movement in these marine mammal outbreaks. Very concerning stuff. Now, not to kind of all doom and gloom on you, it is a bit of a terrible and sad story, and that's what is happening globally. But I'm here in Australia and we don't have this virus. And so what we're going to do is we're going to kind of focus in here onto what I'll call the local scale. I mean, Australia is massive, so I don't know how local it is. Maybe we should call it a country scale. But I'm going to call it local scale because this is Australia scale. So we've been working on avian influenza here for a few years. And unlike the Northern Hemisphere, where kind of dynamics in low path, that's what that LPAI stands for, had really been worked out, it was really unclear what was happening in Australia when I moved here. You know, the virus was here, but no one could really figure out what was going on. People would sample and just not really find a lot. And so we've been doing quite a lot of work in the low path space. And now with a sort of high path risk, we've realized that everything that we've been doing is extremely helpful in informing us on high path avian influenza preparedness and developing appropriate surveillance systems. And so what I've done is I've kind of framed this bit in these kind of questions. So when we came to Australia, there was a lot of things we didn't know. So which species are susceptible to avian influenza in Australia? In the Northern Hemisphere, we know everything is about mallards, but we don't have mallards here. So where does that leave us? How do these viruses arrive in Australia, or are they just kind of stuck here? What drives prevalence of avian influenza in Australia? So like, is it always the same? Is it the same as the Northern Hemisphere? And how are viruses dispersed within Australia? Like really basic stuff. But these were things that really needed to be worked out and are really helpful in understanding what could happen if high path arrives. So in response, we've actually been sampling wild birds for avian influenza for over a decade. My colleague Marcel Claassen actually started a few years before I arrived. And we really focused in on groups of birds that are important in the Northern Hemisphere. So these are the terrariforms. These are things like waders, gulls, and terns, and the anseriforms, as well as a few different birds. So what you're looking at here are the list of birds for which we have at least 50 samples collected. They're organized by their host phylogeny. So you can see the host phylogeny on the left there. And then the different colors are the bird families. And I've sort of highlighted terrariforms and anseriforms. And what you're looking at is percentage prevalence. So the percentage of the samples that were positive for avian influenza using qPCR. Okay, same way you go get a COVID PCR test, we do an influenza PCR test. And then I've added the confidence intervals and sample sizes. So essentially what you can see is the dark green and the light green are the clear winners here. So ducks also really important here in Australia, although not all ducks are equal. Similarly, shorebirds, particularly those in the Scolopacidae, are important. So these are the long distance migratory shorebirds. These are the guys that are migrating from Australia to Asia and back every year. But again, not all shorebirds are equal. Some have much higher prevalence than others. Okay, so this is very useful for us and it helps us really hone down on our surveillance. Terns, apparently not that important. Silver gulls, which are our sort of common gull here, not that important. And some of the other species that we co-sampled, especially in the interior, also don't seem to be important for this virus. So now that we know which birds the virus is in, we can try to kind of work out how the virus, oh, sorry, I have skipped a step. So this was virus prevalence, okay? So that's our PCR test. But we've also done seroprevalence, okay? So we can take a blood test and we can look for antibodies against this virus. And the key advantage of seroprevalence is that we maintain antibodies for quite a long time, whereas we can have a virus for 1 or 2 days and then we stop shedding. So if we sample at the wrong period of time, the birds may all test negative, but if they have been affected, then the seroprevalence would, or the antibodies would still be there. Okay, so this gives us a second measure to understand our host range. And so sort of similar with the left plot, the dark green and the light green are clear winners. The ducks there, you can see that definitely a duck is not a duck is not a duck. And we see some differences in seroprevalence. And one of the really interesting things about this seroprevalence is if you look at the second bird from the top, that's sanderling. That's a small shorebird. You guys have them in the States as well. They're pretty common in places like the Delaware Bay. I've seen them in Oregon as well. From our virus prevalence, we saw, so the left-hand panel, that we didn't have any virus detections in this species. And we could easily say, oh, we just didn't sample enough of them or sample them at the right place at the right time. But if you look at the right-hand plot, the seroprevalence is also 0, okay? That tells us that none of the birds we sampled had antibodies either, which sort of gives us a bit of confidence that maybe this species is not very important in the ecology of avian influenza virus. And interestingly, it's not just in Australia that these birds aren't infected. If you look in Delaware Bay, where pretty much all shorebirds are affected, sanderlings almost always come out with no viruses. It is quite interesting. So the seroprevalence gives us sort of an extra measure to help us be sure that we have a really good understanding of the host community of this virus. Now that we understand this, we can try to work out how viruses can be moved between Australia and Asia, okay? So in most of the world, the story is all about ducks, 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 ducks. Ducks have the highest prevalence, and they are the movers of this virus. But the thing is, Australian ducks are actually endemic to what we call the Australia-Papuan region. So on the map there, you're looking at satellite tracks of these really pretty ducks called grey teals. So, they kind of fly around Australia a bit. They may go to Papua New Guinea. They may go to New Zealand, but that's where they kind of stop. And essentially, in Australia, we have this really interesting thing called the Wallace line. It's a really important biogeographical sort of line. Everything to the left and to the right are very, very different. And that seems to dictate what birds do here too. And we just don't have any duck species that cross that line that go into Asia, which means that it cannot be ducks that are moving these viruses around between Australia and Asia. But of course, we have these long distance shorebirds in the Scolopacidae that do these amazing migrations. They're just so cool. Like these birds, I don't know, they're just a few grams. They're tiny little things. The thing that I always say is that they're the same size as a Tim Tam, which is probably the size of 2 Oreos, maybe 1.5 Oreos. Okay, like they're really little birds, but they do amazing things. So what you're looking at here on the right is a migratory track of redneck stints that have been captured here close to Melbourne. The big circles are all the places they stopped on their way, and off they went all the way to the north of Siberia and back, and stopping all along the way. So these species are the most likely species to be bringing viruses here. Now, we don't only need to rely on sort of bird migration data to understand that. We can actually also look at genomics, virus genomics to look at that. And so, sorry, I'm going to skip that slide. And so I just actually want to show you some contrasting patterns of what happens in ducks and shorebirds. So we have this paper. I'm actually in the process of submitting it this week. So you guys can read it hopefully later in the week if you're interested. And so we've been co-sampling ducks and shorebirds here in Victoria for a long time. And we isolated a number of H4s and we sequenced them, and we found quite an interesting pattern. So when we looked at what was in the ducks, here we go, the viruses we found were similar to other viruses in Australia, and they fell into this lineage here. So what you're looking at here is a time-structured phylogenetic tree. So the tree is kind of put on a time axis there on the bottom. So, you can kind of work out here, if you go here, you go down. You can kind of work out when this event happened. And these are kind of like confidence intervals. Okay, that's the band around the estimate. And so this lineage here in pink, well, it's been in Australia for quite a long time. It entered from Asia into Australia in the maybe 1970s. Okay, so it's been here a long time, and that's where all our duck sequences fell. But our shorebird sequences were in a totally different part of the tree. Okay, they fell into this little clade down here and are actually really similar to sequences from Asia, not sequences from Australia. And we've done sort of some time structured work, and they probably only entered Australia 1 or 2 years before we detected them here. And so this really suggests that shorebirds are moving these viruses. And when we have a look at some of the other genes, we can definitely see how there is connectivity between Asia and Australia in the phylogenetic trees of the genes. So this really tells us that shorebirds have the capacity to move viruses here. Now, you could say to me, Michelle, you're telling us a story about low path, but could shorebirds really be moving high path here? And this is actually a question that we have thought about, and we have used serology to try to understand this. And so what you can do is you can take the blood samples from the birds. We do our first serology test that tells us whether or not there are antibodies against influenza in general. And from those that are positive, we can do a really specific test, taking the antibodies, so the serum, the antibodies, and challenging it with different kinds of viruses to see which ones react. And so we can take the positive antibodies and challenge it against low path H5 they have in Australia, and then a few of the different kinds of high path viruses that we know circulate in Asia. And then we can plot them, okay? So every column here is an individual. White means I didn't have enough serum to do that test, but gray means I did the test and it was negative, but blue means I did the test and it was positive. So in 2013, 2014, we had Ridnack stints that had antibodies in them that reacted to this high path virus 2342, and that's kind of the code for the virus, which was circulating in that time. So I'm just going to open this up. We actually went back to our archives and tested many, many years of stint sera and found that it's not overall, but we do have individuals with antibodies that are reacting to these viruses, which suggests they have been exposed to these viruses in the past. Not in Australia, but elsewhere along their migrations. So if the birds are exposed and survive, that tells us they could potentially be vectors for bringing the virus here, though that hasn't happened yet. So the next question is, okay, so we know what birds it's in and we know how it could get here, but what happens here? Australia is kind of a weird place, and it's a little bit different from the rest of the world. And so I showed you the plot here on the left before. That was actually part of a study wherein we were trying to understand lots of factors. And so we looked at the effect of host phylogeny, of age, of year, and a few other factors on avian influenza prevalence to try to understand what dictates prevalence dynamics in Australia. So in the Northern Hemisphere, there's a really strong seasonal thing happening where every single year prevalence is increased in the autumn or the fall, quite high through the fall, and then kind of tapers off the end of the year. But in Australia, we just don't see that happen. It just seems overly chaotic, but it's not. Okay, so host phylogeny, really important. Age, extremely important, where young birds are more likely to be infected than old birds, which is not surprising because young birds have fewer antibodies than old birds. And the other thing, we also had quite a strong year effect in our data set. So why would we have a year effect? I mean, duck fashion is exactly the same between years. It just doesn't really make sense. But it kind of does if you live in Australia. Australia is really highly influenced by sort of these large ocean systems, such that El Niños and La Niñas have massive, massive impacts here, more so than anywhere else I've ever lived. So the pictures on the right there, that's the same wetland photographed at about the same time of the year. But the top one is in a dry year and the bottom one is in a wet year. And so if you were a duck and you needed to breed, you know, it's dry, the conditions are just not good. Where are you going to build a nest that you can't be seen? What are you going to feed your ducklings? Like, does it even make sense to breed this year? No. So ducks here may not breed every year. There are some years they just totally abandon breeding, which means in those years, there are almost no juveniles in the system. They're only sort of these adult birds. This is in contrast to these wet years. The ducks go, oh my God, this is fantastic. Let's have some eggs, raise some babies. And then it's still wet. The conditions are still good. So these ducks may have a second clutch. They may even have a third clutch in the year for as long as conditions are good because things here are quite boom bust. And so in these wet years, we have a huge proportion of juveniles in our populations, and these juveniles are immunologically naive, okay? And so you can imagine then in these wet years with lots of immunologically naive individuals, we actually have very high influenza prevalence. And in the dry years with almost no immunologically naive individuals in the population, we have almost no avian influenza detectable in the system. And so just to kind of show you that in a different way, this is just kind of a plot of rainfall here in the southeast part of Australia. The blue line there is kind of the smooth rainfall. And we've plotted yearly avian influenza prevalence here. I think this is from ducks. And we kind of see the little dots are much higher in the years where the blue lines are higher, and the dots are much lower in the years when the blue lines are lower. You know, in the last few years, it's been La Nina years. It's been excellent for us. We have isolated loads of viruses from the ducks. Things are going really well. But, you know, in the years prior to that, we really struggled to get anything out of the birds. So it's actually sort of this large scale climactic conditions that are important in the system, but we detect that through a year effect. Now, this is important for ducks, but interestingly enough, it's also important for poultry systems here in Australia. We have homegrown outbreaks of highly pathogenic avian influenza here. So these viruses from ducks enter poultry systems, they emerge here, and then they're stamped out or controlled through control measures. And these outbreaks in poultry happen in or slightly after wet years. And it's not because the dynamics are affecting what's happening in poultry. It's just that we see such a big buildup of the viruses in ducks that the risk of introduction of a virus into the poultry sheds is much, much, much higher. And so that's when we find outbreaks. So understanding what is happening in wild birds and driving wild birds is actually really important for us for risk management for poultry and to clarify why these poultry outbreaks here seemingly happen quite randomly and hard to measure, but it's because they're linked to rainfall, because that's what's happening and they're driving what's happening in the ducks. And then the last point is how are viruses dispersed within Australia? And Australia is a weird place. So where you guys are, you have really predictable cycles of migration, okay? You can look at where the geese are and they should be in the same place in their migratory cycle every year. So birds move up, birds move down. In Australia, sure, the shorebirds are doing predictable patterns of migration, but most of the endemic species here don't migrate. They are nomadic. They're essentially following around water. So wherever it rains in the landscape, birds kind of show up. I don't know how they do it. It's really incredible. Like it can rain in the middle of the desert, and one day later, all these ducks will be there. How they do it, I don't know. But it means we can't really predict where viruses are moving or how they're moving around the landscape. And so we've tried to look at this a little bit with virus genomics. And so how we do this is we build sort of these trees in Beast. Here on the left is an H7, okay, and we can kind of identify all our different locations. And so we can then take our map on the right, and all the locations that occur on the left, so the different colors, are plotted as white circles there on the right. And then we can ask our sort of software to say, can you measure migration events between these different locations? And so the gray lines on the map on the right there means there could have been a migration event there, but one was not recorded. And then the blue arrows show you where migration events were recorded. The color is the number of migration events between those locations, and the arrow is the directionality of the migration event. Okay, so here migration event just means transition between locations. And so we've done that now for a bunch of different subtypes. Because they're so diverse, we'll analyze them separately. And essentially, we see really good connectivity between the three states here on the bottom right, or in the southeast part of Australia. That's not surprising for two reasons. One is we have a major river system kind of on the border with Victoria, New South Wales, and South Australia. Here, ducks don't understand boundaries, so they kind of come into this system and go out again. So we should see connectivity there. But the reality is also that that's where most of the viruses in this study come from. Okay, these are the states with the most resources for this work, have the longest history of doing avian influenza. So it's the states for which we have the most data. So makes sense, doesn't make sense. If we look at some of the internal segments, okay, so the HAs we have to analyze separately because they're so diverse, but they mix and match their other segments, which means that we can actually lump way more sequences together in our analysis if we look at some of the other internal segments. And we see a far more complex picture if we have more data. So again, most of the arrows, very, very dark here in the southeast part for the same reasons as I explained before. But we do see a lot more connectivity between the south and the north. So we see kind of virus movement between and across the north. It's not all north to south, and a bit across east to west. So there is some connectivity. I think this exercise was an interesting exercise to start. And what I tried to really press upon policymakers is for this exercise to work, we need a lot more genomes. We need a lot more surveillance because it can be extremely useful and informative, but we're not quite there yet. So let's see. Oh, I have to go a bit fast. So, switching scales again, we've been talking about populations in the last two sections. Now, I wanted to think a little bit about the individual scale. I'm not going to spend a lot of time on this only because, well, it's interesting. Not a lot of people do work on here, and I have some things to show you, but no real conclusions. It's mostly just kind of an open question, okay? So we generally work on populations. This is a plot from the study site in Sweden. It's 10 years of data. What you're looking at is virus prevalence in the population. So what happens is that almost no influenza in the system, winter and spring. Then what happens is all the ducks go on the landscape and breeding. There's lots and lots of ducklings. And then once the ducklings kind of get big enough to fly, it's autumn, it gets cold, all these immunologically naive birds come together to congregate and migrate, and we see high levels of influenza prevalence in the autumn, okay? That's what's happening at the population. But what is happening at the individual level? So we did think about this a little bit. So when I was in Sweden, we had this great big massive duck trap. Uh, and it's a really effective trap. We could catch 100 birds a day quite passively. The ducks were essentially lured into the trap because there was a lot of food. But also we had 10 female ducks that were living inside the duck trap. That we called them lure ducks because they were used to lure the male ducks into the trap. And once the bold males go into the trap, all the other birds will kind of follow. And it occurred to us at one point we should just be sampling all these liver ducks because they're going to be infected by their conspecifics. And here we can follow individuals. So here are our 10 individual liver ducks. This is time and day of the year. So I think this starts in September, if I'm correct, and it kind of goes through the autumn and the fall. White means the birds were negative. Gray means the birds were positive for influenza, but we couldn't isolate it, so we don't know what subtype it is. And all the other colors are the different subtypes that we detected. So there are some patterns across all these individuals. Like we see a lot of red and blue in the beginning, and we see a bit of orange and green in the middle. But there are also certainly differences, okay? So some birds shed for a long time. So that bird on the bottom compared to the one right above it have really short, discrete infections. You know, all these ducks were purchased from the same breeder. They're the same age. They're the same strain. Genetically, they should be really similar to each other. But, you know, we have stochastic processes of infection here. This bird here, number 5, just kind of seems to shed intermittently for ages and ages and ages. Again, this is low pathogenic avian influenza. These birds are clinically healthy, but they have a really large burden of infection. So different patterns in virus shedding. We also looked at antibodies, so just the general influenza antibodies. So above the dotted line means they're positive for antibodies, below means they're negative. And so, you know, we saw that in the spring when we started sampling them again. Most of the birds had antibodies. In the summer, it dipped. So the birds are laying eggs. So there's a trade-off here between sort of growing eggs and maintaining antibodies. And so most of them lose antibodies. And then they're all rapidly infected again in the autumn and go up again. But look at this green line on the top. This bird just keeps all its antibodies. It's different from the rest. Or this purple one down here below. Okay. That one, it just never crosses the line. So, you know, this is one of the very few papers looking at individual effects. I think it's like one of 3 because it's very hard. And I think there's a lot of opportunity here, but we haven't pursued that. I also decided to dig out some data that I haven't published, that's sitting on a hard drive that I have intention to publish one day. So just, you know, this one is not for Twitter if you're tweeting all this, because it's very unpublished and raw data. So this is from Ruddy Turnstones on King Island, these really amazing migratory birds. We've been sampling them twice a year since 2013. The last time I updated this plot was a few years ago. I apologize, but I had to dig deep for this one. And so at a population level, very little virus, except we have kind of these occasional outbreaks. And then the bottom part here is seroprevalence, so the proportion of population that has antibodies. So something interesting happening here at population level. But then I went to look at the individual level for the individuals that we have resampled. So this is a pretty cool study site. We can resample individuals. And a large proportion of the birds are always negative. So white means no sample, gray means negative. This light blue color means it was right at the sort of threshold between negative and positive, but we'll call them negative for here. We have a small proportion of birds that are always positive. We have some in which we see seroreversion. So they were positive in the beginning and then they become negative. So they lose those antibodies. Others that seroconvert. So they were negative in the beginning, but become positive, presumably because they've been infected. And then we've got a few birds that are in this transition. They're positive one time, negative the next time, positive the next time. So I don't have any conclusions other than to show you this is something we are thinking about. You know, and it's hard to make generalizations from individual levels. I think sort of follow-up studies looking at immune responses and things like that would be really important. But this is a very unexplored scale, I think, for avian influenza, and one that should be thought about more. So I'm just going to try to tidy this up. Sorry, it's already 1:45, and I definitely have too much stuff in here. But we've been talking about multi-host single pathogen through this story, and I just want to change the story a bit to multi-host multi-pathogen or multivirus, because the reality is influenza is not the only thing that's in these birds. Influenza is one virus as part of a bird community. And so I started thinking about this a little bit in my PhD. And so one of the years from a duck trap in Sweden, we looked at influenza. That's the black line there. So this is day of the year. So again, this is the autumn. This is virus prevalence. The black line is prevalent, modeled prevalence, and the dots are the confidence intervals. And then we also screen these ducks for coronaviruses. So birds are also affected with coronaviruses. This is a coronavirus that's in the family gamma coronaviruses. So COVID, SARS-CoV-2 is beta. This one is gamma. It's a coronavirus that's commonly found in ducks, recently named duck coronavirus. And we also looked at avian paramyxovirus 1. So that causes a disease in chickens called Newcastle disease. It was one of the first bird viruses ever isolated. It was isolated about 100 years ago. Okay. So look at this. We looked for 3 viruses in our ducks, and lo and behold, they're all there. Then when I was looking at this data, we found quite a lot of birds were co-infected. Okay, so birds were positive for more than one thing. You see this kind of bimodal shape again, because that's reflective of the dynamics of the system. But almost all the co-infections involved influenza. So AIV is influenza, okay? And so you see a lot of black and green and gray. Those are all the co-infections that involve influenza. And there's only like 2 blue ones here that don't involve influenza. So what is going on here? We really thought about this. Influenza must be quite important in the system. And so what a colleague did, which was really interesting, is we decided to model the prevalence of the coronavirus with and without taking into account influenza virus prevalence. Alexi's a pretty smart guy. I don't remember the details of how he did this, but essentially the red line is higher than the black line here. So prevalence for taking into account influenza is higher than not taking it into account. And the conclusion is that there might be some synergistic effect here. And that's kind of as far as we took it at the time. But when I started working with metagenomic data, moving here to Australia, this was like the first thing that I wanted to do. I wanted to reinvestigate the effect of influenza. So what we're doing here is we're using RNA-seq. We're essentially pooling together 10 individuals captured at the same place, same time, same species, doing RNA sequencing on those samples, which is essentially sequencing everything, okay? And then pulling out all the viruses that are in that sequence data, which means we're not limited to viruses we've described before. We can literally find anything that's in there that can be found through a BLAST search, not a BLAST N search, but like a amino acid search, okay? And so what I did is I had 2 libraries from ducks and 2 libraries from turnstones. One library I knew had influenza in it because I selected samples that were influenza positive, and one that I knew were absent, so I selected samples that were influenza negative. And so this is just the virus abundance, okay? It's the proportion of the reads that are viral. And you can see that the 2 columns on the right are higher than the 2 columns on the left, okay? So the avian virus abundance is higher in influenza positive birds compared to negative birds. And that's even when we remove the influenza reads. So something interesting is happening. We can then go and have a look at what the viral reads are. So again, it's kind of laid out the same. We have the two duck libraries, the two turnstones, negative, positive, negative, positive. White means that virus genus is absent. The color means it's present, okay? And it's on a scale, a log scale there. So the light colors means it's present but scant. The blue means it's present. There's quite a lot of it. And then the stars mean I pulled out a full genome, okay? So what you can very quickly see is that there's more colored boxes in a darker shade with more stars in the positive columns than there is in the negative columns, okay? So we have a higher load and diversity of viruses in libraries that also contain avian influenza. Super cool. We also looked at beta diversity, okay, which looks at how similar the libraries are to each other. And we put it on this MNDS plot. And sort of if the dots are kind of close to each other, that means the composition and abundance of the libraries is really similar. And if the dots are far apart from each other, it means they're quite different. And this was the plot the reviewer wanted, but we actually have another plot where the two influenza triangles are almost on top of each other. So just a slight difference in the analysis. Okay, but essentially we had a statistically significant signal here that influenza was really important, and that the libraries containing influenza were more similar to each other than all the other libraries that we looked at in this study. So influenza is definitely having an effect on this system, but I don't think anyone has really followed this up, and I think I really would like to come back to it because I don't really understand exactly what's going on, other than there is definitely something going on. You know, we've found this trend sort of through two really different approaches in different study systems. But yeah, really sort of interested to think about this a little bit more. This is thankfully the last slide. I'm going to stop talking very soon. Instead of writing a really nice conclusion, which I normally do, I just thought I would stop talking and show you some really nice pictures of some of the fieldwork we've done here in Australia. We have some really pretty ducks here. I mean, mallards are lovely, but we have some really interesting ducks here. And recognize that all the work I've talked about has been done with lots and lots of people. Their names are all up there. And the work here in Australia is funded very much by Department of Agriculture, Fisheries and Forestry and the Australian Research Council. And yes, I think I'll stop talking now. So thank you. Yeah, and happy for any questions.
For questions, if people just want to unmute, they can, or if they put them in the chat, I can read them out, whatever is preferred.
I'll start out with a question. Oh, sorry, I didn't raise my hand. Yeah.
You go first, Dan.
Okay, that was a great presentation, super interesting. I thought the metagenomic work was also really, really interesting at the end. And I have just kind of a low-hanging fruit question, which is it possible that the samples that contained flu and other viruses were just sort of better quality samples, and that's why you picked up so many viruses, and then the other ones were sort of didn't have enough RNA or weren't from quite the right anatomic spot or anything like that?
So the quality of the samples was the same. So essentially, yeah, I do a lot of quality control to make sure we can definitely compare between the libraries. So it's the same RNA concentration that was sent, and all the samples were diluted so that, you know, the pools were constructed exactly the same way. So that's definitely something I thought about when I started designing these metagenomic studies, because I didn't want that to be the key reason. But one of the things that I have been thinking about in the back of my head is maybe inadvertently we selected more juveniles because they're influenza there, but I don't have that much age data on all of the birds. So for example, for ducks, you can age them as juveniles in the autumn, so for 4 months, and then you can't age them anymore. They all look the same. So it could be that we've just kind of accidentally done that, but we don't know that. But I don't know, maybe it is that there is something interesting happening with influenza there. I think there is a lot of opportunities to follow up on this with other approaches. But yeah.
Great, thanks.
So my question was actually very similar or about the same topic as Anne's, except I wasn't thinking about any of the kind of the technical aspects, but was thinking more about any ideas that you may have maybe in terms of immunosuppression or something like that that could then allow other viruses to come in. So for example, you see this a lot in honeybees, for example. They have tons of different viruses, and when they have deformed wing virus, for example, you're more likely to find other viruses. And so I wondered if you surely have thought about it, and it would be interested. You mentioned it's of interest, and it sounds like you want to go into this. So if you were to, what would be the first thing that you would really want to figure out?
Yeah, so influenza has an antagonist, a protein that is designed to push down on the immune system. And interestingly, in avian influenza, there are two really different versions of this, whereas in human and swine and equine influenza, there is only one version of this. So I think that there must be sort of some really effective way of suppressing the immune system. But, the problem with doing immunology on non-host model species is that it's very, very, very hard. I've got one colleague in the world that is doing it. She's based in Canada, actually, and she has spent her whole career doing it. And I feel like she's gotten this far just because she's the only one and it's very hard. So yes, I'm pretty sure in the discussion of this paper, I wrote about the likelihood that it's probably that this sort of NS is really good at suppressing the immune system, creating an opportunity for other viruses. But I think like you'd have to do a proper immunology study to really understand. You'd have to do an experimental infection, having birds, inoculating them with just influenza, and a few other things, and then trying to look at the immune expression patterns. Yeah, lots of opportunity. I'm hoping one day maybe I entice her with this and she's really keen to do it. Yeah.
Otherwise, you can come and do it here at Emory. Lots of people
I could.
interested in it.
Yeah, look, I have loads of ideas on lots of things I'd really love to do. So I'll just kind of tack that onto the list, maybe one day.
Thanks.
Really great talk. Thanks for covering so much ground in such a, nice way of tying it all together. In the big duck trap in Sweden, that's a really fascinating set of follow-up. But when you had the thing over the time, watching the nesting season in the middle, is there some behavioral aspect to that? Do your duck decoys behave differently, interact less with some of the males coming in? Or I'm just wondering if there's a component to that may be behavioral rather than immunological in the development of eggs and so on.
Yeah, I mean, it is behavioral because they're kind of spending a bit more time on land relative to the water. But how the trap is that compartment shares, it's like a mesh between the wild ducks and the lure ducks. And it's the same water that's on both sides. So even if the ducks are behaving, spending a bit more time on land, they're still definitely spending time in the water. And there's still a lot of droplets and transmission happening. And we put feed on the water side of the cage for both the wild and lure ducks, kind of forcing them to hang out in the back there. So there's definitely a physiology thing that is, when they were laying eggs and making eggs, we saw this massive drop in antibodies. It could also be behavioral. So, but in the summertime, when they're laying eggs like that, we actually don't have a lot of influenza in the system in general, not in the wild ducks and not in the lure ducks. It really starts again in like late August and September when the wild birds are starting to congregate and migrate. We see these really big peaks in influenza, and that's when all those antibodies essentially shot back up. That's when influenza re-entered that system. But again, it's an interesting thing that we found because it had never been shown before, because most people don't resample wild birds to be able to follow antibodies like that. And so I think we're the only ones that have really observed that. And it's fodder for lots of interesting follow-up studies. It kind of in that framing of trade-offs, and immune trade-offs.
Great, thanks.
Yeah, thank you.
Hi, I had a question. That was a really great talk. Thanks. I was just wondering if you could talk a little bit about either what's like known, or if you could speculate a little bit about how actually transmission is happening amongst these like marine mammals in South America, or like between birds and mammals. Like I'm kind of, I think I understand at least a little bit like how transmission of influenza works in humans and as a respiratory virus, but yeah, like what's happening in the ocean that's like moving these viruses between species?
Yeah, all right. So we'll start with low path because that's what's happening everywhere. That's what's co-evolved. That's what's going on in wild birds. Okay, so we mostly find these viruses in water birds, and transmission is fecal oral. So we have a duck that's in shallow water. It's infected. It's defecating in the shallow water, and all its other friends are feeding where it's been defecating. Okay, so the water has lots of viruses in it, and the birds are being infected. We have these ducks in Australia, they're called pink-eared ducks, and how they forage is they essentially form like a little circle, one bum to head, and they go in circles as they forage. And so guess what? Prevalence in these birds is really high because you just follow the leader, right? One has influenza, it'll just affect the whole chain. No. Okay, so that's low path, that's what's happening. High path, so interesting. I think all the rules have been broken. It's so bizarre. So we definitely have fecal oral bird to bird. That is definitely happening. High path is a bit different from low path in that it can infect birds everywhere. So low path is a GI tract infection in wild birds. In high path, we're getting both the GI tract and respiratory tract, and we're seeing brain infections as well. So fecal oral is happening, bird to bird contact is happening, feathers is happening, and birds sort of making secretions on each other is all happening at the same time. The mammals, okay, so most mammals are getting infected by eating wild birds. That's just largely what's happening, and they're almost all getting these terrible neurological infections. And so the marine mammals, there's been outbreaks in seals in the US, for example. And so what people originally proposed is that marine mammals, are definitely eating sick and dead seabirds. There's photographs of it. They're, free meal's a free meal, especially for a sea lion. But the other thing is, if you think about haul outs of where pinnipeds are hauling out, there's almost always cormorants and seagulls and things hanging out in the same places. And there's always a lot of guano in these places. Like, you ever been to a rookery? It smells real bad. Okay. And so one hypothesis is that these animals are rolling around in feces of birds, and that is how they're getting infected, is they're being infected by the birds around them. And that all made sense until we started seeing 40,000 of them. Like, this just doesn't really make sense anymore. There was a lot of neurological animals, but we also started seeing these respiratory tract infections. And so my colleagues in South America strongly believe that there is mammal-to-mammal transmission, and it could be through respiratory secretions of these animals that are having the respiratory signs. Don't know. Could also be that the animals are having gastrointestinal infections and their feces are in colonies and they're infecting all their other friends. Like, I don't know if you've ever seen southern elephant seals. They like lie in these pools of fetid water that other animals have crapped in and peed in, and they're just like lying in there, really. It's disgusting. It's so disgusting. That kind of makes sense. The other interesting thing about the elephant seals in Peninsula Valdez, so this is in Argentina, we saw essentially all the pups from that year die, almost all of them. So hypothesis there is milk or vertical transmission. But again, just don't know, my colleagues in South America, there's just 2 of them that are responding to all of this. So, I think they kind of showed up at this colony and everything was dead. So they drew some conclusions, but they're not really sure. So I think around transmission, there's loads and loads of questions. I think there is probably a lot in the works at the moment. I wouldn't be surprised if all kinds of people are doing ferret experiments with all kinds of viruses that have been collected to try to work this out. So I would argue more questions than answers, but answers are in the works. The last thing I'll say is about the dairy cows, because everyone keeps asking me about dairy cows. Again, more questions than answers. Question number 1 is, how did it go from birds to cows? Option 1 is in the US. You guys feed cattle poultry litter. Very bizarre. It's illegal everywhere else in the world, but you guys are doing it, and there's a lot of avian influenza outbreaks in poultry in the same places as cattle. So potential number one, unclear, unverified, we don't know, but could happen. Number two is that grain and food is often stored in big barns. They don't necessarily have great biosecurity. We know birds are going in and out of those barns. So we could imagine that birds have also contaminated the feed here. Or three, poultry dairy houses are open. Also, again, birds coming into those areas. So all of that could have played a role. All speculation, nobody knows. Cow to cow, USDA is saying that it's mechanical. Okay, so what we're seeing in cattle is mastitis, infection of the udders. And I don't think they clean the suction cups between cows and dairy barns. Okay, so we're essentially seeing mastitis go from cow to cow to cow. And how it's spreading between states is that almost all states that have positive cases have received cattle from Texas. So we think it's just that it's happening in Texas and being spread around. The cats in the barns make sense if it's mastitis. So if it's milk that is having high virus loads and there's cats in barns, that makes sense. And the human infection, if the guy has conjunctivitis. Okay, so my theory, which is just my theory, is that he was dealing with milking equipment and he got stuff on his hands and he rubbed his eye. Yeah, but I think that all questions, lots of hypotheses, we will see what they come out with. Yeah, I think that covers all the transmission possibilities. Maybe I forgot one, but I think that's it. Yeah.
Thank you so much. Unfortunately, we are out of time. Sorry, Elizabeth, if you wanted to say thank you.
Well, this is my fault. I talked too long. I put too much in the talk. I'm sorry.
No, this was really great. I really loved how you framed the whole talk and everything. Thank you so much. It was awesome.
Yeah, well, thanks for having
So-
me, and feel free to email if you got any questions.